So we're going to talk about today. We're going to talk about membrane potential. So first, we're going to go over concentration gradients. So when you're going from a high concentration to low concentration, we call it passive transport, and this just happens slowly, right? And it happens on its own. Active transport is when you're trying to go against your gradient. So first, let's talk about some types of passive transport. So here we have simple diffusion, which is with your small and nonpolar molecules. Osmosis is like simple diffusion but of water. So water, hypotonic is water in, hypertonic is water out. Facilitated diffusion uses membrane proteins for large or polar molecules. Okay? And a lot of people, aka me, get confused with why facilitated diffusion isn't active transport. And that is because when you have active transport, you require ATP for energy. So in facilitated diffusion, you're not using ATP, nor do you have an ion gradient. So some examples of active transport is we have primary active transport, which is direct ATP use, aka your Na plus and K plus pump. Then we have secondary active transport, which uses a gradient of another solute. So this is your symport and antiport. Next, we have endo and exopsychosis. So endo meaning bring in is when the membrane brings in material. So when we use pinna, so that's just for fluids. Phago is for solids, and then exopsychosis, vesicles fuse with membranes and release contents. Okay, this you guys should know, but I feel like your biology teachers probably have went over this in detail, so we should all be okay. So next we're gonna go over chapter 8.4, which is all about specialized membranes.